This is the Chapter 12 recording. We're starting with slide 28 in the Chapter 12 PowerPoint slides, and we are now going to cover the process of protein synthesis, or the two processes involved in protein synthesis are transcription and translation. So this is slide number 28, and you can see that coding for a protein starts with a DNA molecule. So a piece of DNA that codes for a protein is called a gene. So we start with our gene, and then one of the strands of the DNA serves as a template strand, and this happens in the nucleus. A process called transcription occurs, and that is when the template DNA strand serves as the model to code for a strand of RNA, and we call that messenger RNA. So we say that the DNA is transcribed into messenger RNA. And you can look at the DNA code and tell what the messenger RNA code is going to be, except for, and you can't see it on here because they didn't give a good example, but RNA does not have thymine. It only has uracil. So anywhere that there would be a T in the RNA code would be a U. And I know you can't see an example of that here, but you just have to remember it for now. So for a transcription of this particular template DNA strand, thymine is going to be complementary to adenine. So T pairs with A, C pairs with G, G pairs with C, C pairs with G, T pairs with A, and then the last four are G's and they pair with C's. So the messenger RNA is divided into sequences of 3 nucleotides, and each sequence of 3 is called a codon. So we have 3 codons in this particular messenger RNA. That messenger RNA is going to leave the nucleus and attach to a ribosome. And a process is going to occur called translation. So each of those codons will be translated into amino acids, and those amino acids will join to form a polypeptide, and the polypeptide will eventually become a protein. So we actually have a chart that tells us which codon becomes which amino acid. So this codon AGC, let's see if we have a chart that I can show you. Yes. All right, so on this messenger RNA codon chart, we find ACG, which is right here. It must have been AGC, sorry. Yeah, AGC codes for serine. So let's go back. And that's our first amino acid. AGC codes for serine. And then GAC codes for aspartate or aspartic acid. I've already forgotten. I'm sorry. I think it was GAC. Aspartate or aspartic acid here. GAC, pretty sure. Yes, and then the last codon is easy, 3 C's. So we find CCC, and that is proline. Make sure that's what they have. Yeah, this is the amino acid proline. So serine, aspartate, and proline is the amino acids we would get from the translation of these 3 codons. Now, in the process of protein synthesis, there are 3 types of RNA, and we talked about messenger RNA takes the message from DNA in the nucleus to the ribosomes in the cytoplasm. But there are 2 other RNAs. There's ribosomal RNA, or rRNA, which forms the ribosomes. There are 2 that form the 2 ribosomal subunits which make up ribosomes. And then there is transfer RNA, and that actually transfers the amino acid to the ribosome, to the correct spot where it belongs in the protein. And it is determined by the code in the messenger RNA. So structure of RNA is a little different from DNA. It is composed of nucleotides, but it's only single-stranded. And also, instead of the nitrogenous base thymine, RNA has U, or uracil. Okay. Here is a chart that shows you the differences between RNA and DNA. The sugars are different. The sugar in RNA is called ribose. In DNA, the sugar is called deoxyribose. They are both 5-carbon sugars, and both of them have a shape with 5 sides, a pentagon shape. So we call them pentose sugars. The bases in RNA and DNA are the same except for thymine. Thymine is found in DNA. Uracil replaces thymine in RNA. RNA is single-stranded, DNA is double-stranded. RNA does not form a helical structure, DNA does. All right, so the genetic code consists of codons, which are found on the messenger RNA. They are 3 nucleotides make up a codon. Each codon is specific for one of the 20 amino acids. And we know amino acids are the building blocks of proteins. There are 64 possible codons, so that means there's more codons than amino acids. So the codons can be redundant or degenerate, and that means that there is more than one codon for each amino acid. Each amino acid has 2 or more codons. But the codons are not ambiguous, which means that each codon codes for only one amino acid. But there can be more than one codon for each amino acid. So, for example, let's find one that has like a lot. So leucine. Leucine has UUA, UUG, CUU, CUC, CUA, and CUG. All of those can code for leucine. Proline is one that has, I see 4 different codes here. There also are codons for stop. So UAA, UGA, and UAG mean stop making protein. They'll be found at the end of each sequence. And then there's a start codon, which is always AUG, and it always codes for the same amino acid, which is methionine. AUG is the only codon, by the way, that codes for methionine. And methionine is always the first amino acid in every protein. So the genetic code is universal. That means with few exceptions, all organisms use the code the same way, have the same genetic code. It works the same way. It's a sequence of 4 nucleotides, A, C, G, and T, and they code for proteins. The same 20 amino acids are found in all the proteins for any type of organism, any species. And the same 64 codons from the codon chart code for the same amino acids. So starting with transcription, we're going to cover that process. One difference in DNA replication and transcription is the enzyme that adds the nucleotides to form the messenger RNA is not called DNA polymerase. It is called RNA polymerase. So in transcription, you still have a DNA molecule that has to be unzipped, and one of the strands is used as the template strand. One of the parental strands is the template strand. And that template strand is used to code for the messenger RNA strand. Now here you have an example. Let me make this bigger. You have an example here that shows where normally in DNA replication, we would see A pairing with T, but RNA does not have thymine. It has uracil in place of thymine. So everywhere there would be a thymine is going to be a U. So T still is going to pair with A, G will pair with C will pair with G, A will now pair with U. But that is where you're going to get the nucleotides to make the messenger RNA transcript. And then the messenger RNA transcript is going to undergo some editing. And the editing is adding a cap on the 5' end. That's just the beginning of the molecule. And then adding a poly A tail, in other words, many adenines. 150 to 200 adenines on the 3' end, or the end of the molecule. The beginning of the molecule is called the 5' end, and the end is called the 3' end. So a cap is added, a tail is added, and then there are pieces of the pre-messenger RNA that are spliced out or that are removed, and then the pieces that are left are spliced together. So the final messenger RNA is even shorter than the original gene from the DNA. So the exons are pieces of mRNA that are going to remain, that are going to be coded for the protein. The introns are not. So the introns will be removed, the exons will stay, and the exons will be spliced together. The removal in eukaryotes is done by spliceosomes that contain small nuclear RNAs, snRNAs. And the ribozyme is the enzyme that's made of RNA rather than protein, and that is what cuts the introns. So here is your DNA. After transcription, you have introns and exons in the messenger RNA. You have then the addition of the 5' cap, which is a modified guanine, the addition of the 3' poly A tail, and then you have the removal of the introns, and the only thing left is the exons. The spliceosomes will splice them together. And then you have your final messenger RNA, which is going to be shorter than the original DNA piece or gene. There is a section that tells you there's a possible function of introns, but I want to move on to translation. So translation is going to involve the messenger RNA attaching to a ribosome. Let's just find a picture of it. The messenger RNA attaching to a ribosome, and here's what a ribosome looks like. It has a large subunit and a small subunit. The messenger RNA attaches to it, and then the transfer RNAs, each transfer RNA contains an anticodon that is complementary to every codon on the messenger RNA. And it will also contain the amino acid that matches up with that codon, with that messenger RNA codon based on the codon chart. So let's look at this. Oops, sorry. I'm trying to blow it up, not make it go away. All right, so the first amino acid that is transferred to messenger RNA is always methionine. The codon is AUG. So the transfer RNA brings methionine to that spot because the transfer RNA contains an anticodon, UAC, that is complementary to the messenger RNA codon. So that's how it knows exactly where to land or where to attach to. Okay, so that occurs in the, I guess you say the A site. I always say A site is the amino acid site. That's where the amino acid is added. And then the messenger RNA will shift and move to the P site, move that same codon and that same amino acid to the P site, which is where this, I always think of P as permanent. It means polypeptide, but I always think of it as this is where the amino acid becomes a permanent part of the polypeptide. So for me, the A site means added. That's where the amino acid is added to the polypeptide. And then the P site is where the amino acid becomes a permanent part of the polypeptide. Then the messenger RNA moves over, but the amino acid stays. So the messenger RNA moves to the E site or the exit site, and the transfer RNA exits, and then the amino acid stays. So the messenger RNA basically keeps shifting to the left as it adds more amino acids. And we can see a picture of that. All right, so you can see here a transfer RNA has come in and brought the valine to add to the 4 amino acids that are already there. So valine and the transfer RNA with the anticodon CAU are attached here to GUA, the codon, and that is in the P site. So The permanent amino acids are methionine, serine, alanine, tryptophan, and valine. These are abbreviated, but these are 5 amino acids that have become permanent parts of the polypeptide. Now you're going to see in this next step that the anticodon and the transfer RNA carrying the next amino acid that's going to be added come into the A site. And then you're going to see that aspartic acid, I think is what it is going to be, and it's added in the A site. So in the final picture, you see that the entire messenger RNA shifts to the left to the P site, and the aspartic acid is the 6th amino acid that's now a permanent part of the polypeptide. The previous messenger RNA codon moves over into the E site, and the transfer RNA for that with the anticodon exits. Okay, so that just keeps happening like over and over and over until you have your entire polypeptide, until a stop codon is reached, until a stop codon is encountered, and that means stop making protein. Okay, so UGA is one of the 3 stop codons, and that tells the ribosome to stop making protein. And then that protein is made. Now we know the protein is just a polypeptide that has the primary shape of the amino acid chain, and now it's going to have to undergo shape changes and have a secondary, tertiary, and possibly quaternary shape. But that happens later.